The goal of this lecture is to talk about how our biochemistry, our human physiology changes during exercise. And exercise actually causes our body to move away from homeostasis. That is to say, our oxygen levels start deviating from an acceptable range. Glucose starts leaving from the acceptable range, what have you. So in no way am I suggesting exercise is a bad thing, but during exercise, our body needs to maintain these specific variables, whether it's oxygen, glucose, blood pressure, what have you, within their acceptable ranges, or even elevate those in the case of high intensity exercise, whether we're talking anaerobic or aerobic exercise. So one of the first things we need to look at is insulin. And we've talked about insulin in previous videos. We've talked about insulin release from pancreatic beta cells. This image right here is not showing pancreatic beta cells. This image is showing glucose uptake at our cells downstream of the pancreas. That is to say, insulin, being a hormone, is going to be released from the pancreas into the bloodstream, and then it's going to bind to insulin receptors at specific tissues. The primary targets for insulin in the body are the liver, skeletal muscle, and adipose tissue. Tissues like the brain are insulin independent. That is to say, they do not require insulin for glucose uptake. Now, the liver requires insulin, but it's not really for glucose uptake, and we will touch upon that. So let's just assume this cell right here, the large cell right here in blue or purplish, is a skeletal muscle cell. The function of insulin at skeletal muscle cells or at adipocytes is to facilitate glucose uptake. That is the movement of glucose from the blood plasma into those cells. Now, truth be known, skeletal muscle doesn't absolutely require insulin, nor do adipocytes. But with the insulin stimulus, they are permitted greater glucose uptake because they have more glucose channels embedded in their membrane. What we see over to the left here is a secretory vesicle within the skeletal muscle. And embedded within the membrane of the skeletal muscle, embedded within the membrane of that secretory vesicle are GLUT4 channels. These are merely glucose channels that aid or facilitate glucose diffusion into the cell. But those channels need to be embedded within the membrane of the cell, and that's what we see over here to the right. So via the process of exocytosis, the membrane of the secretory vesicle fuses with the membrane of, in this case, the skeletal muscle fiber to implant those GLUT4 channels, which allow glucose to move from an area of high concentration in the blood plasma into the cell. That's the function of insulin. So if we take a look at insulin, and to be clear, there's two hormones that regulate glucose within the blood. One is glucagon, which we see on the left. The other is insulin, which we see in this bottle right here. Insulin is secreted from pancreatic beta cells. Glucagon is secreted from pancreatic alpha cells. During what's known as the fed state, that is after a meal, we have elevated blood glucose. We have an increased concentration of glucose within our bloodstream. The elevated levels of glucose, as we discussed many weeks ago, are going to stimulate pancreatic beta cells to release insulin. So during elevated blood glucose levels, insulin is secreted. The whole idea is when we have elevated blood glucose levels, glucose is out of the acceptable range that we want within our blood plasma. Insulin is going to decrease blood glucose levels by facilitating glucose uptake into body cells. During the fed state, parasympathetic activity is going to stimulate those beta cells to release insulin. And it's not only parasympathetic activity, it's going to be the increased blood glucose levels. So in tandem, elevated blood glucose levels and parasympathetic activity to the beta cells will stimulate the release of insulin. Insulin is going to facilitate glucose uptake by embedding those GLUT4 channels in the plasma membrane of those cells. It's also going to facilitate or increase glucose metabolism. It will stimulate glycogenesis, that is the formation of glycogen. Glycogen is the storage form of glucose. It is a molecule made up of monomeric subunits known as glucose in this case. It is also going to facilitate, that is to say, insulin is also going to facilitate lipogenesis, the formation of lipids like triglycerides, and it will also stimulate the process of protein synthesis. One thing I want to reiterate once again that we see down here in the corner, the brain is insulin independent. So since we just talked about insulin, it's worth our while to mention glucagon. Once again, glucagon is released from pancreatic alpha cells. During the fasted state between meals, when we have lowered blood glucose concentrations, glucagon is going to be released from pancreatic alpha cells to stimulate a number of things. The first that we see here is glycogenolysis. Glycogenolysis is the breakdown of glycogen into the monomeric subunits, which are glucose, and putting that glucose into the blood plasma so our body cells can use it. Glucagon also stimulates the process of gluconeogenesis, which is the production of glucose, or glycolytic intermediates is probably a better way to say it. The production of glycolytic intermediates from non-carbohydrate precursors or non-carbohydrate substrates, like we looked at previously, for example, with glycerol, which enters the glycolytic pathway at glyceraldehyde 3-phosphate. Glucagon also initiates the process of ketogenesis, which produces ketone bodies that the body can use for energy. One ketone body will produce upwards of 22 ATP molecules. Okay, so let's take a look at sympathetic activity and hormonal influences on our muscle cells during exercise. Sympathetic activity inhibits the secretion of insulin from pancreatic beta cells. I'm going to say that one more time. Sympathetic activity is going to prevent the release or inhibit the release of insulin from pancreatic beta cells. And that may seem counterintuitive because during exercise, our skeletal muscle fibers are going to need glucose to make ATP so they can do the work to create force and tension within skeletal muscle fibers. But the key here is that contracting skeletal muscle fibers will induce the exocytosis of GLUT4 channels in the cell membrane of skeletal muscle. That is to say, if we look back at this image right here, embeds these GLUT4 channels in the cell membrane or plasma membrane of skeletal muscle fibers. That's when insulin is bound to those cells. This happens in skeletal muscle fibers in the absence of insulin, and it is merely due to the contraction of skeletal muscle fibers or skeletal muscle cells. So muscle contraction embeds those GLUT4 channels into the sarcolemma or plasma membrane of skeletal muscle fibers. And why is that cool? Because without insulin being released from pancreatic beta cells, other cells in the body are not going to be able to uptake glucose or use that glucose as readily as they could if insulin was available to help facilitate or increase their glucose uptake or their use of glucose. And because those cells can't use it as easily as skeletal muscle fibers, then the majority of that glucose is going to be going to the skeletal muscle fibers that are trying to do work during exercise. If we look at the hormones glucagon, cortisol, epinephrine, and norepinephrine, they all promote glycogenolysis, which is the breakdown of glycogen, once again, to increase blood glucose levels. Cortisol and epinephrine, norepinephrine, also promote lipolysis, specifically the breakdown of triglycerides into glycerol and fatty acids. And as we discussed previously, the fatty acids via beta oxidation will be converted to acetyl coenzyme A and enter the Krebs cycle, and glycerol will enter the middle of glycolysis. Okay, so let's talk about glucose and the liver. What I said previously is that insulin acts primarily on the liver, skeletal muscles, and adipose tissue. And it does so with adipose tissue and with skeletal muscle by facilitating glucose uptake via those GLUT4 channels. In the liver, specifically in the cells of the liver known as hepatocytes, insulin does not facilitate, nor is it needed for glucose uptake. During the fasted state, there is low insulin. And during the fasted state, there is glucose secreted from these hepatocytes due to the breakdown of glycogen, otherwise known as glycogenolysis. During the fed state, during high insulin levels, glucose moves the other way into the hepatocytes. So to be clear, during the fasted state, glucose is leaving the hepatocytes to increase the blood plasma concentration. During the fed state, during high insulin levels, glucose is moving from the blood plasma into the hepatocytes, into the liver. Insulin is not needed for that to happen. But what insulin does is to increase the production of hexokinase, which is an enzyme that converts glucose into glucose 6-phosphate. Here we see some hepatocytes. Glucose will enter the hepatocytes and be converted to glucose 6-phosphate via the presence of the enzyme hexokinase. Insulin will stimulate or cause an increased rate of production of hexokinase, which will convert glucose immediately into glucose 6-phosphate upon glucose's entry into the hepatocyte. Once again, insulin is not needed for glucose to enter the hepatocyte. But if glucose is immediately converted to glucose 6-phosphate, then it's going to maintain glucose's concentration or chemical gradient, favoring it to move into the cell. If glucose is converted to glucose 6-phosphate or into glycogen, the concentration of glucose within the hepatocytes remains low. And if the concentration of glucose within the hepatocytes remains low, glucose continually moves into the liver, into those hepatocytes. To be clear, it is insulin acting upon increased production of a hexokinase that converts glucose into glucose 6-phosphate. In the liver, insulin has no impact or does not embed GLUT4 channels into the cell membrane of the hepatocytes. Okay, let's talk about VO2. VO2 is the amount of oxygen used by muscles to produce ATP. It is known as oxygen consumption, and it is measured in liters of oxygen per minute. Whereas VO2 max is a person's maximal rate of oxygen consumption, which turns into being an indicator of the ability to perform endurance exercises. Once again, VO2 max is the highest rate of oxygen consumption attainable during maximum exercise for an individual. And a VO2 max and VO2 is going to vary from person to person. To be clear, VO2 max is not the exact same thing as aerobic capacity. Aerobic capacity is the ability to take in oxygen, to transport it by the cardiovascular system, and then to use oxygen by the skeletal muscles to do aerobic work via the production of ATP. VO2 max is merely the highest rate a person can consume oxygen during maximal exercise or maximum exercise. So at the start of exercise, oxygen consumption increases so rapidly, that is to say, oxygen consumption from the skeletal muscle increases so rapidly that it is not immediately matched by oxygen supplied to the muscles. Once again, during the beginning of exercise, oxygen consumption by the skeletal muscle is greater than oxygen supplied to the muscles. So initially, ATP to nourish or to provide energy for the muscles comes from the residual ATP that we've talked about in creatine phosphate. Then aerobic metabolism kicks in via the oxygen stored in myoglobin and hemoglobin. And then as exercise continues, the cardiovascular system and respiratory system catches up. Then there will be a continual production of ATP as long as that exercise is aerobic in nature and the body can sustain itself during a given amount of time or intensity. But after exercise, we have to repay all the debt we've incurred, all the oxygen that we've used. We need to replenish that. That oxygen is used to metabolize lactic acid. It's used to restore ATP and creatine phosphate. Keep in mind, oxygen is needed to make ATP. And I need to restate that it's certainly not needed. ATP can be produced anaerobically via glycolysis, but much more ATP, as you all know, can be produced via the aerobic pathway. So the oxygen debt is going to be recovered via aerobic metabolism of glucose. This is also going to replenish the myoglobin oxygen levels stored in skeletal muscle. Okay, so we all know what it means to hit the wall where we can't work out, we can't sustain endurance exercise any longer. And this is due to the limits of muscle activity. That is to say, how efficiently our muscles are working and utilizing oxygen. There are limits to our cardiovascular system and to our respiratory system. One big thing with skeletal muscle is the amount of mitochondria. Mitochondria, which are the organelles within cells that produce upwards of 32 ATP in the presence of oxygen. That's not including the ATP produced during the Krebs cycle or glycolysis. But that muscle activity is heavily dependent on the amount of mitochondria. And some people are born with more mitochondria in their skeletal muscle fibers than others. That is to say, it's genetically determined how many mitochondria individuals have. But through exercise, an individual can increase their efficiency of their mitochondria. The mitochondria will work more effectively to produce more ATP over time. Another significant factor for skeletal muscle are the amount of blood vessels, and exercise can increase blood vessels going to skeletal muscles. That's a process known as angiogenesis. The cardiovascular system is aimed at only one thing, and that is transporting oxygen to the tissues. A better trained heart is going to deliver oxygen more efficiently, and it will not have to work as hard. And the respiratory system is more genetic in nature. That is to say, the maximum capacity lungs can hold. That also can vary with exercise, but a lot of it is based on genetics. So just to reiterate, our cells have mitochondria, which we see right here. And the mitochondria produce ATP. Some individuals have more mitochondria than others. There's nothing we can do about it, but we can make the mitochondria with exercise, with regular endurance exercise, aerobic exercise, can make those mitochondria more efficient. Angiogenesis is the production of more capillary beds feeding that tissue, and in this case, skeletal muscle fibers. So exercise will facilitate the process of angiogenesis. And then, of course, the cardiovascular system and the respiratory system play a part. The limiting factor in endurance exercise tends to be the heart. At maximum exercise, the heart is hitting a wall much sooner than the lungs are hitting a wall. Okay, but let's talk more about the lungs. What regulates breathing during exercise? And the three things are receptors and proprioception or proprioceptors. Mechanoreceptors and proprioceptors are detecting changes in movement or body position, which will send signals to the respiratory center in the medulla oblongata to increase the rate and depth of respirations. Another signal for the respiratory center are the chemoreceptors that are detecting levels of carbon dioxide, pH, and oxygen. Diminished levels of oxygen send a signal to the respiratory center in the medulla oblongata to increase the rate and depth of respirations. Increased carbon dioxide and a decrease in pH will do the same. And potassium. Potassium, it has been shown that increased potassium concentration in the extracellular fluid will increase the rate and depth of respirations. And the theory behind that is the more action potentials there are during exercise, more potassium is going to be released in the extracellular fluid in the process of repolarizing those cells. And that's going to activate another set of chemoreceptors in the carotid artery, sending a signal up to the medulla oblongata to increase the rate and depth of respirations. Okay, let's talk more about cardiac output. And as we recall, cardiac output is heart rate times stroke volume, the amount of time a heart beats per minute, and the amount of blood that is ejected from the left ventricle after one ventricular contraction. Cardiac output is generally 5 liters per minute. That is to say, your total volume of blood over the course of a minute is ejected from the heart. During well-trained athletes, it can be 6 to 8 times more than that. That is to say, in a well-trained athlete, cardiac output can be as high as 40 liters per minute, and that would be during exercise, of course. Heart rate is modified by sympathetic activity. So as we recall, parasympathetic activity reduces heart rate to about 60 to 80 beats per minute. Exercise is first going to cut off parasympathetic activity, and that's going to raise heart rate to about 80 to 100 beats per minute. And then if sympathetic activity kicks into gear, heart rate is going to be greater than 100 beats per minute. So that's 1/2 of cardiac output, the amount of times the heart beats per minute. But then how is stroke volume modified? One is force of ventricular contraction. Sympathetic activity, in addition to impacting the sinoatrial node to increase the rate, sympathetic activity is also going to increase the force of ventricular contraction and hence increase stroke volume. Venous return can also increase stroke volume, but it gets a little bit complicated. So venous return, the more blood returned to the heart upon skeletal muscle contraction. Keep in mind the skeletal muscle pump facilitates the movement of blood back to the heart during exercise. There's more contraction of skeletal muscles. So in theory, there's going to be more muscle moving back to the heart and moving into the left ventricle and the right ventricle as well. And the greater stretch in the myocardium during the return of blood to the ventricle is going to stretch out the myocardium and theoretically increase force of contraction up to a point. That point is what's referred to as the Frank-Starling principle, which we've talked about previously. Deep inhalations will also facilitate the movement of blood back to the heart. But increase heart rate during this same time that the skeletal muscle pump is working and the respiratory pump is working, the increased contractions, or I should say the increased heart rate, is going to limit the amount of blood moving into the ventricles via the skeletal muscle pump or the respiratory pump. Because if it's beating very quickly, there's a limited amount of time for that blood to return to the ventricles. Okay, let's talk about increased blood flow to the muscles during exercise. So I have vasomotion up here, and there's two aspects to vasomotion: vasoconstriction and vasodilation. During exercise, arterioles going to skeletal muscles vasodilate, where arterioles going to most other tissues in the body, not including the brain or the heart, are going to vasoconstrict to allow more blood flow to the skeletal muscle. At rest, skeletal muscles receive about 20% of cardiac output, but during exercise, they can receive upwards of 90% of cardiac output. In skeletal muscle, there's going to be a decrease in oxygen concentration, an increase of CO2, and a decrease in pH, and an increase in temperature. And we've elaborated on this many times with the cellular respiration equation. All of those factors will lead paracrine activity at the skeletal muscle fibers to further dilate blood vessels to allow a greater and greater flow of blood to those muscles.